This is drawing 42 on T cells. We just learned how B cells work. They are the ones that make all those little antibodies. T cells do not make antibodies. They have a very different function from the B cells, although they're very related because they're both lymphocytes. To understand how T cells work, we first need to learn a little more about how body cells work. Cells are like little houses with no windows. Okay, here's your little, you can imagine there's a little neighborhood here. Our cute little cell neighborhood. Now they would have doors, right? Imagine portals in and out, I guess, but no windows. So, if there's something bad going on inside a house, maybe there's a burglar or something, a thief running around, or maybe the oven's on fire or something, and there's no windows, you can't see inside, it's all going on inside. How are you going to know what's going on? How is the neighbor going to know what's going on in this house? Well, cells have a way to tell each other what's going on inside. So we're going to see how they communicate what is going on in this space here. We're going to draw a little section of body cell. So it's not, we're just going to see a section of it rising up like this. Make sure you leave like an inch, inch and a half, couple centimeters up here, okay? And we're just going to draw like this. Real lightly. Now we're going to have to erase a little bit of it. Go lightly. This is kind of your guideline. And it doesn't, if it's not perfectly round and everything, that's okay. It'll be just fine. Okay, and then we're going to draw a second line underneath this because what we're drawing here is the outer membrane. And it's always good to remember that the plasma membrane is a phospholipid bilayer. Remember, it's got all those little balls with tails. The tails would be all in here. Okay, so this is this bilayer, 2 layers of phospholipids. Okay, so that's our outer membrane. And then we're going to draw the endoplasmic reticulum. Okay, that's one of those cell parts that a lot of students are kind of vague on. You know, it's fun to be able to say endoplasmic reticulum, but, you know, say, what does it do? And people kind of stop. Golgi bodies, yes, they package. They're like a warehouse. They're like a post office. You know, we kind of know the mitochondria is the powerhouse of the cell. What does the endoplasmic reticulum do? We kind of go, wow, I can't remember. So here's one important thing the endoplasmic reticulum does. Leave a little bit of space in here and then come down, draw a guideline like this. So remember, the endoplasmic reticulum is like reticulum, reti means net. It's like this huge network of tubes, basically. It's a network of tubes. And remember, the ER is connected to the nucleus. The nucleus has those 2 phospholipid layers, and the outer one is actually part of the endoplasmic reticulum. Okay, so it goes. So we're not seeing the whole thing. This is not the nucleus, and this is exaggerated. Okay, this is, if this were the size of the cell, this thing would be a lot smaller. So this is not to scale. But to be able to understand what's going on, we're going to make the ER very large, and we can label this ER. And this is also plasma membrane. You can draw a second little line here. So it's sometimes hard to remember what the ER is made out of. It's just a tube, plasma membrane like that. Okay, and then if you want to write endoplasmic reticulum, you can, but I'm just going to put ER. And then I'm going to put a little cell coming in the picture right here. Just peeking in. This will be that police cell, or whatever you want to call a, maybe it might be a killer T, we'll talk about those, or an NK, we'll get to those. So here's the police cell that's coming to check. And I'm going to put a little receptor thing. We'll see what these are. So this is kind of like the cell's eyes. Remember, cells can't see. They have to feel. And these are going to be the feelers on this police cell. It's going to come along and feel what's on the edge of the body cell. Okay, and of course what you call them receptors. Receptor is a very general word for just about any of those little knobby things on the outside of a cell. Okay, so this says the body has a roaming police force that constantly scans for trouble. The police will kill any cell that cannot prove that everything is okay inside. Body cells must clean house and cover their outer membranes with samples of the proteins that are floating around inside. If the cellular police detect an intruder or a sickness, an intruder would be maybe a virus, sickness maybe a cancer or something like that, they will kill the cell. So there's really not a hospital for cells once there's something bad going on. If the cell is sick, the body's way of handling it is you just kill that cell and grow a new one, okay? Because a cell is not an organism in and of itself, okay? You're not killing not killing the organism. Cells are expendable. They're designed to live a little while, die, and you know, new ones come to take their place. That's just the way it is in bodies. So let's see down here. I'll zoom in a little bit. We're going to start this way in the picture, and there's numbers. Let me show you down here. This section, what is going on in the picture, and we're going to coordinate our picture with these numbers. Okay, so we're going to start with number 1, and I'll read them off. But while we're drawing, you won't see these while we're zooming in, but that's what we're doing. So number 1 is going to occur right over here. So this is where number 1 will be. And number 1 says, there are always proteins floating around in the cytosol. So let's just draw some little random proteins. Where might these proteins have come from? Well, maybe the lysosome digested some stuff. Maybe it's proteins that are waiting. They were floating around and waiting to be used somewhere in the cell. Maybe they're proteins that enzymes that its cell is using. Okay, so as long as they're normal proteins that belong there, it's okay. If there's an intruder inside, then these proteins won't look. There'll be bad proteins floating around. So number 2 says a tiny organelle called a proteosome acts like a shredder and chops these proteins up into tiny pieces. So we're going to draw a little protein shredder machine, and it looks like a little tube. So draw a little oval, a couple lines over, like that. You can make it look round if you want to. Okay, and it really does look like this. It's this little barrel-shaped thing, and the protein structure really does look like a little tube. Okay, so this is called the proteasome. Proteasome. It's printed down there in the words if you want to spell it right. Okay, so some of these proteins are going to go into, now remember, it doesn't know what it's doing. It doesn't have a brain. It just goes around, and any protein that gets sucked in here will get shredded. It's like a paper shredder, and out the other end will come little tiny bits. How tiny are these bits? Well, they're usually about 5 to 10 amino acids long. That's pretty tiny. So now we got a bunch of little tiny shreds floating around. So let's make this number 2 right there. So this is the little recycler unit. Then, you know, these little protein bits, 5 to 10 little amino acid chunks, they could get picked up and recycled, made to another protein. This is what takes care of proteins that are folded incorrectly. You know, you have these spell checking, editing machines going along, looking for mistakes in DNA copying, looking for mistakes in protein. And if there's a protein that's folded wrong, just wasn't quite right, it'll go into the shredder and get chopped up into little tiny pieces, and then the little tiny pieces can get recycled again. So you've got these little tiny pieces floating around. And then there's a little portal here. This will be number 3. And just draw this little portal. We can also, if you want to darken up, I think this will be good along here. Number 3. The little pieces, some of them float into, they just kind of, they don't know to come over. It's just some of them will happen to be near there, and they'll get sucked in and pushed into the ER. Okay, so we don't have anybody lost yet, right? There's little big pieces of protein floating around. They get chopped up by the little shredder into little pieces, and they go into the endoplasmic reticulum. So meanwhile, another thing is going on right here in the middle here. We're going to make a ribosome sitting right on the outer edge. We can make the 2 subunits, the big one and the small one. This is our ribosome. And then what does the ribosome do? It takes messenger RNA, so we'll make a little ticker tape. Remember, messenger RNA looks like kind of half a DNA. It looks like or half of a zipper. So I'm going to put rungs on one side, and I can label it mRNA. And the messenger, it would be much longer than this. Mine's short, but the messenger RNA has the instructions for how to make a protein chain. So some of these little amino acids, transfer RNA is going to be bringing these little amino acids over, feeding them into the ribosome, and when the protein chain comes out, now sometimes the ribosomes are floating around, right, in the cytosol, and when they make a protein, it's just right there. But sometimes there's a little code right on the messenger RNA that says, take me to the ER. Put me inside, and this is one of the ones that has the code. It's very important that this protein is spit into the ER. Otherwise, it would be useless. If it didn't have that little code on it, it just wouldn't work. So there's our little protein going into the ER, and this protein is going to need to be folded up. Okay, so let's label this number 4. Meanwhile, the ribosome is making a polypeptide and spitting it into the ER. Then what happens is this has to get folded up, right, to make its appropriate shape. And when this gets folded, I'm going to show you a picture of what the protein looks like. Kind of a computer generated. This is the closest we can get to an actual picture. I mean, it's hard to take pictures of atoms and molecules, right? But this is one of those things where you can see the alpha helixes and the beta sheets and things. So you notice it's got 1, 2, 3, 4. So like a purple, a pink, a green, and a kind of a greenish blue here. And then there's this crazy little thing sitting in here. So they simplified this shape. They've thing here, thing here, thing here, thing here, and a little kind of U-shaped thing with a piece of something sitting in there. So they decided this is too difficult to draw, so they simplified it and made it look like this. So it's much more understandable, isn't it? Okay, so actually this one, this is MHC2. Let's look at this one. So here's this protein, this protein, this protein, and this protein. And then there's a little piece of the protein that's going to coil down and go through the plasma membrane. So you can really see here they drew all those little circles and lines, and you can see the little lollipop molecules there. All right, so you can simplify it even more, which is what we're going to do. We're going to make ours look sort of more like this, okay? you can just basically make it a rectangle with a little opening at the top, except that you can tell that these are, it's hard to tell in the picture, but these are two different, this one and this one are different. And it's got this one little stick that's going to go down and stick in the plasma membrane. And what it does is it functions as a clip. There's the part that sticks in the membrane, and this is the part that functions as like, it's be like a clothespin or something. You could stick something in there and it'll hold it. That's exactly what it's going to do. So we're going to make ours look like, just focusing in there. We're going to make it look like just, if you want to make these still kind of curly-Q, like proteins, something like that. Okay, and then you remember that there's still, like over here, there's little proteins floating around. Okay, well there's some over here too, little protein bits. And one of them is going to get stuck. It's going to get trapped in there. You don't have to use color. I was just going to put a tiny bit in there. One of those little protein bits is going to stick right in that clip. Okay, so now we have this little thing here. It's called MHC1, and in this picture there's two of them. I'll show you. Okay, there's MHC1 and MHC2, and it's easy to remember. MHC1 has one stick and MHC2 has two sticks. Otherwise they look pretty similar, right? So we'll find out about MHC2 in the next drawing. So this thing called MHC1, which we're going to take a close-up look at in just a minute at the bottom of the page. Right now, let's find out what goes on here in the rest of the membrane. It comes over here to this little section of membrane. I'm going to need to get a little erasing there. And the ER, its job is to create vesicles. Now it's really good at doing this because basically the ER is just this huge tube of plasma membrane, and a vesicle is just a bubble made out of plasma membrane. So it creates this little bubble, and this little clip thing goes up, right? Draw it again, scribble, scribble, there we go, and it create the little protein in there. So it goes into this little bubble, and that's number 6. Whoops, number 5 here. We need to do number 5. This is where it gets folded into the right shape. 5, 6. And then this little bubble floats off, and we're going to show this little vesicle coming into contact with the plasma membrane. All right, we're going to show it just starting to touch, so you can still see that it's a vesicle. Put the MHC1 inside there. Okay. Floats to the surface. What's going to happen as soon as it touches? It's like this is made out of the same stuff. Okay, the plasma membrane on the outside and the plasma membrane of the vesicle are made of the same things. And it's like two oil drops touching. If you have a bunch of oil drops in water and two of the oil drops touch, as soon as they touch, whoop, they just like merge together, and now you have one bigger droplet. So that's what happens here. As soon as these touch, they kind of join. So we'll draw a picture of what it looks like when they're slightly more joined. The vesicle, suddenly you have something like that. And we'll erase here. It's going to do this. We still have the little MHC1 inside here. So we can put a 7 right here, because what's going on is the vesicle goes to the plasma membrane and joins with it, And this is, of course, called exocytosis. Right? And when it joins, this bubble opens up. So the next picture, you know, the bubble, little vesicle is going to be disappearing. It's straightening out. It's just becoming part of the membrane. Oh, in fact, we should draw this stick going down into here. I think it might be already joined to the membrane. If you just draw the MHC1 stick sticking into here, so we can kind of tell that it's. I mean, a lot of pictures they do kind of show it floating, but it kind of makes more sense if it's already attached to the side. I'll just make mine a little bit longer like this. Okay, and here it is. It's already attached. Just makes more sense, doesn't it? Okay, and then here it is. And then finally, there it is, sticking on the outside. The wrinkle disappears. And there it is on the outside of the cell. And there's the sample. And this is MHC1. All right, and we could label this in here if you want to. Body. Whoops, sorry about that. Losing you. Okay, there you go. Body cell. Alrighty. Let's back out a little bit now. Sorry, I got a little focused in too much there. So let's add a little note right here at the top. We have a little bit of space. Now I'm going to talk about the word MHC in just a minute, but let's, for right now, don't worry about it. MHC1, this little thing, has a double function. Number one, identifies cell as, quote, self. Number two, presents sample. So it's doing two things. It also acts as that little identity tag that says I belong to the body, and it presents a sample. It does both jobs. And let's just write over here about the sample. Let's write a little note. The sample presented on MHC1 should be a normal cellular protein. Okay? Abnormal proteins alert T cells that there is something wrong with the cell. Okay, you can pause that if you need to catch up. I'm going to go back down here. We're going to go down to this section down here. Okay. And we're going to take a close-up look at this MHC thing. We're almost done. This is a short drawing. Okay, this is going to be super, super easy. We just want to draw two of these things. Almost kind of reminds me like of a popsicle. If you've ever seen those popsicles that have like two sections and then they have two sticks coming out the bottom. MHC2 kind of reminds me of a popsicle, except it has a bite out of the top. So this one is going to be, we're going to divide it in four because there's 4 proteins. Put a stick down here. He's got only one stick and a bite. This is MHC1. And then here's the little protein sample sticking out. And this is MHC2. Just like that. And he's got 2 sticks. So MHC stands for Major Histocompatibility Complex. Histo means tissue, major means important, compatibility means compatible, gets along, complex means a complicated little group of proteins, a group. So this group of proteins is all about tissue compatibility. Compatibility with what? Well, with other people. These were discovered when they were doing transplants, right? And sometimes the transplants worked and sometimes they didn't. They didn't know why, and they found out it's because of these things. It turns out, we'll put a note on the bottom here. Let's add this note. Each person themselves has about 50 types of MHC1s. Just when you think biology is complicated enough, it gets a little more complicated. There can be slight variations in the twists and coils and the number of amino acids and things in here, so that there's different shades of MHC1. So each person has about 50 types of MHC1s. These are what they try to match in transplants. So they say that are you compatible, hopefully maybe a parent or a sibling will be more compatible, but the odds are maybe somebody you're not related to just happens to have more of these 50 types in common. So this is what they're checking for. They're looking at these guys, the MHC1s. Let's just put a note here that these are, add a little sample protein here. And this one will look a little different. These are sample proteins. And put a little bracket. This part is what T cells see as self. That's what they're looking for. They're going to check, because those will see the sample. The sample is sometimes self, sometimes not self, but this is the part that they're going to check, this block here. And of course this stick right down here, we can put in this part here, embeds in membrane. It's very mechanical, isn't it? Stick to hold it in, and then a clip at the top, and this is just to be, then this is the identity part. So it's a very efficient little molecule. Okay, and then we're going to see MHC2 in the next drawing and how important that is in the immune process.